Hello, and thanks for being here today. I'm Gerard Santiago, Head of Enzyme Engineering in Nostrum Biodiscovery, and today we'll talk about in silico enzyme engineering, from in silico enzyme engineering to PluriZymes. Before going into details, let me please introduce our company, Nostrum Biodiscovery. Nostrum Biodiscovery is a young startup born in 2016 in the Barcelona Supercomputing Center and IRB, to top research institutions in Barcelona with economical support of Fundación Botín. In Nostrum Biodiscovery, we have two main goals. From one side, we perform drug discovery services. From the other side, we work in enzyme engineering, focusing in both enzyme optimization and the design of our own enzymes, specifically the PluriZymes. We will talk about them later. But the main core of Pele, it's the technology. As I said, we're an in silico modeling company, so our technologies are a key part of our DNA. And our flagship technology is called Pele. Pele stands for Protein Energy Landscape Exploration, and it's a Monte Carlo algorithm, really fast, and it has been tested through many academic and industrial projects. And it's the perfect software to tackle molecular recognition problems. Why? Because with Pele, we combine the random movements of the Monte Carlo algorithm with protein structure prediction, so we can explore the whole binding event. This means that we place the ligand outside the system and we freely explore the overall of the protein without any bias in the simulation until we find the active site and we have an enhanced sampling of the active site binding. So we can perform this type of whole experiments in just hours, something that with molecular dynamics will take weeks of calculation and is far more precise than docking. Let's see in a short video how Pele works. As you can see here, the ligand is freely moving around the protein. Also notice how the backbone it's also moving. And once the ligand it finds the active site, it's capable to go inside and place in almost the perfect identical position as the crystal reference that we have inside. Also notice that when the ligand is closed, the surrounding area, the surrounding side chains also adapt and minimize according to the arrival of the ligand. But let's see how we apply this technology in enzyme engineering. And let's go through a really recent example that will be soon published in ACH Catalysis, where we expand the substrate scope of ester hydrolase. But before talking about how we can expand a substrate scope, we have to discuss what defines a substrate scope. Why an enzyme is promiscuous? It can be answered also through Pele, combination of Pele and other structural parameters. When we took to really different esterases, one really promiscuous, in this plot the red one, and one really specific, the blue in this plot, and we simulate them with Pele, we can see that there are many different behaviors. For one side, we can see how a promiscuous enzyme goes to deep binding energies, and also the substrate goes to really low SAS. But in the non-promiscuous one, the behavior is the opposite. When we move this to the three-dimensional level and we check the overall shape of the cavity, we can see that the cavities are also really different. We have a big isolate cavity in the promiscuous enzyme and a small and surface exposed cavity for the promiscuous one, for the non-promiscuous one. So promiscuity can be summarized in a relation, in a balance between available space and solvent exposure. So to have a promiscuous enzyme, you need a big isolate cavity. Of course, this example is with only two, but when we move this to a big data set of 150 esterases, and we try this idea towards all these different esterases, we obtain this correlation. This correlation between what we call the effective volume, a ratio between the volume of the cavity and the solvent accessible surface area, and the overall substrate promiscuity. And we can see how we can classify perfectly promiscuous enzymes of non-promiscuous enzymes. Then with this information, we can now also engineer enzymes to go from this part of the table to this part of the table. How? Playing with big residues in the active site, so we can open a space and create a cavity, as we can see here. We took this really non-promiscuous enzyme, introduced mutations in those three residues, create this cavity, and simulate the system with Pele, comparing with a wild type as we have here. We have the triple mutant 1, and we can see how the triple mutant 1 behaves really different than the wild type and in a similar way as the promiscuous enzyme. So according to our simulations, we have created a promiscuous enzyme from a non-promiscuous one. But what in vitro validation said? In vitro validation said that yes, we create a non-promiscuous a promiscuous enzyme from a non-promiscuous one, and this promiscuous enzyme that we have here is capable to hydrolyze more than the substrates than the previous one, the wild type, but we lost activity. Why we lost activity? The reason was here in position 16, that it's nearby. Position 15 is a catalytic serine that would replace for a glycine. This shorter residue allows the serine to have high mobility, something that we were able to identify through molecular dynamics, and instead we replace it for alanine and leucine and try both and create a new generation. This new generation leads us to TM2, triple mutant 2, that has an expand substrate scope, or really expand substrate scope, but also keeps the wild type activity, and also in some cases expanding. So when we try also different combinations like double mutant 2, where we just keep those two mutations and let the wild type, we also have an expansion of the substrate scope and a remaining of activity, proving the key role of this residue in the maintenance of the architecture of the structure. And now let's talk about PluriZymes. As you remember for the first slide, PluriZymes are our way to design our own enzymes. But what is a PluriZyme? A PluriZyme is an enzyme with two active sites. One of them introduced artificially in the monomer. How we introduce these active sites? Using Pele. Using Pele, and as we said also before, we can explore the surface of the protein in a Maya simulation until we obtain not only the active site binding, but also non-catalytic areas, places where the ligand feels comfortable, but non-catalysis can happen. So it has been a classical strategy in enzyme engineering to put bulky residues in these areas, so these areas get closed and you enhance the population of your area and enhance activity of the enzyme. So instead of this more classical approach, we introduce the necessary mutations to create a second active site. We start with ester hydrolase for two main reasons. Our collaborators in this project were key experts in this family of enzymes, and also the catalysis of ester hydrolysis only requires from water from the bulk as a cofactor, and the rest happens through amino acids. So no need of big cofactors of metal binding sites. And we did this in 4 different enzymes: 2 esterases, 1 LPMO, and a really recent results that came from the past 2 weeks, also in the transaminase. Let's see results in esterases. I put here the example of one of them. For all the enzymes I mentioned, the PluriZyme approach works. And in this example, we have the original exploration where we map the original active site, but also identify this potential area. And there we introduce the necessary mutations to create the active site, and lines again the simulation aiming for these catalytic positions we see here. In this other plot, we can see how the binding now it's modified, and this big undefined area it's defined and creating a really good minima because we are creating an active site. When we move this to in vitro validation, we obtain not only activity but also really interesting results. In blue, we have the wild type enzyme. In red, in green, sorry, the enzyme, when only the artificial active site is working, and in red, the active site, the enzyme with both active sites are working together. So when both are working together, we have additivity. We have also expand the substrate scope of the enzyme, and the artificial active site is so exactive as the wild type. There is no main difference between the activity between both the artificial one and the original one. Then, once we have this, we went one step further and create two-step reactions in the same monomer. How? Using 3-site inhibitors. These 3-site inhibitors bind covalently in the native active site, modifying this native active site to a chemocyte. Then the artificial one keeps doing the original natural reaction, and the covalent inhibitor is introducing a new one. So we can have two-step reactions. So far, using these covalent inhibitors, we have set up Michael addition, Fiedel cap reactions, and proteolysis together with the Hester hydrolysis. As I said also before, we have the LPMO results. In an LPMO, we want to challenge the approach towards a totally different architecture, a different type of folding and a different type of mechanism, and introduce there an artificial esterase site. Why? The aim for this project was that LPMOs that work towards cellulose fibers, when they found esterification in the cellulose fiber, they got inhibited by those esterifications, and introducing artificial esterases on the edges can clean these esterifications before they can inhibit the LPMO active site. We perform a similar approach as we showed before, but focused on the edges of the active site, of the edges of the surface, of the binding surface, due to the mechanism of the LPMO. The LPMO lands in the cellulose fiber and move in the cellulose fiber. So we were focused on those two areas. In this area, we introduced an artificial esterase that is capable to hydrolyze esters in glucose monomers and small dimers. So we think that this new model that now it's patent pending can overcome this limitation in the LPMO industrial application. Finally, and just as a reminder, we are offer fee-for-service for activity enhancement, directed evolution. Also, we can discuss about PluriZyme design as a service, and recently, thanks to the technology we're now setting up, quick thermal stabilities. Thanks for your time, for your attention. Please feel free to ask any questions right now, or feel free to contact me through my email or phone number or LinkedIn. Thank you very much.